Welcome back, future medtechs. In this video, we'll dive deep into one of the most essential processes in biology. Also known as the protein synthesis. Get ready to uncover how your cell turns genetic blueprints into life-saving proteins. Because without translation, there's no action in the cell. While we got some time, why don't we review some of the foundational topics that will help us understand our new ones? This game is called Central Dogma Relay. I'll flash a few questions on the slide and I'll give you guys some time to answer. Are you guys ready? What enzymes unzip the DNA strand? What is the answer? Correct! It's the helicase. Which strand does the Okazaki fragments occur during the DNA replication? What's your answer? Correct! Your answer is lagging strand. You guys are masters at this already, but today we're going to tackle the last step of central dogma, which is But what is it exactly? Before we tackle extensively, let's go through the intended learning outcomes. First, define the process of translation. This learning outcome must include identifying the key components involved in explaining their role in the process. Second, describe the main stages of translation, developing our cognitive skill in outlining the sequential steps of the process. Lastly, explain the relationship between codon sequence and amino acid order. Take a moment to look at your hands. The bone, skin, muscles you see are made out of cells, and each of these cells contains many millions of proteins. How are these proteins made in cell? For starters, the instructions for making proteins are written. In a cell DNA, in the form of genes, it undergoes replication, transcription, and lastly, translation. Translation is the last step and central dogma of molecular biology, which flows in the DNA, RNA, and protein. While transcription copies the genetic message from DNA into RNA, translation is a part where the message is read and converted into a chain of amino acids, a protein. This occurs in the cytoplasm at structures called ribosomes, which act like molecular factories. For translation to happen, we need the several key players. First one is the RNA, the message carrying the genetic code. The second one is the tRNA, the adapter molecule that brings the correct amino acid to the ribosome. The third one is the ribosome, is the machine that reads the message and builds the protein. The fourth one is the codon, groups of 3 nucleotide bases on the mRNA. The fifth one is the anticodon, 3 base sequences of tRNA that matches the codons. These all work together to form a protein, like a team building a structure from individual bricks. Let's clear up the difference between a codon and an anticodon. A codon is a sequence that 3 nucleotide bases found on the mRNA. It's like a code word that tells the cell which amino acid to add next during the protein synthesis. On the other hand, an anticodon is also known as a sequence of 3 bases, but it's found on the tRNA. Its job is to match the codon on the mRNA through base pairing, like a puzzle piece to ensure the correct amino acid is delivered. Think of it this way. Codon is a message, and anticodon is the decoder. Together, they make sure that the right pieces are added to build the protein, accurately and efficiently. Every 3-base codon in an mRNA specifies 1 amino acid. There are 6 possible codons, but only 20 amino acids. So some amino acids have multiple codons. A codon is a group of 3 nitrogen bases nucleotides on messenger RNA or mRNA. Each codon corresponds to 1 specific amino acid, which is the building blocks of proteins. Let's try a simple given this RNA sequence. AUG GCU, UAC. Using a codon chart, AUG equals to methionine, CCC equals to proline, and UAG equals to stop codon. So these resulting proteins would start with methionine, proline, and then the end of the trend. As a review, to check your understanding, in all of those processes, where does the translation process occur? What's your answer? Correct! So we all know our cells are making new proteins every second of the day. And each of the proteins must contain the right set of amino acids linked together in the right order. That may sound like a challenging task, but luckily, our cells are up to the job.
Okay, so before we proceed, may I ask if everyone can hear me just fine or can hear me? Please give me a thumbs up or a heart react. Okay, I can see that most of you can hear me, so let's begin. Now, before we proceed, do any of you guys know what the main stages of translation is? Can anyone answer? Anyone? Sige, ako na lang. So, there are actually three of those stages. We have initiation, elongation, and termination. Let's start with the initiation. In initiation, the ribosomes attaches to the 5' end of the mRNA. What's mRNA? That's right, messenger RNA. And scans for the start codon, AUG, which this codon always codes for methionine, which is one of your 20 essential amino acids. So every protein begins with methionine. Once the start is found, the first tRNA, which binds to it, and the large ribosomal subunits joins in, and the process is ready to begin. Now, after our initiation stage, we have the elongation stage, where the ribosome moves along the mRNA one codon at a time. Each codon is matched by a specific tRNA with the correct anticodon and amino acid. As tRNAs bring amino acids in, the ribosomes link them together with peptide bonds, now forming a growing polypeptide chain. This continues until the ribosome reads a stop codon. Now, after our polypeptide linkage is formed, we now have the termination stage when a stop codon, which are UAA, UAG, or UGA, is reached, no tRNA can match it. Instead, a release factor binds and the ribosome lets go of the polypeptide chain. The ribosome disassembles and the newly made protein begins folding into its final shape. Now that we are done tackling the main stages of translation, let's have a quick recap. I will be asking three questions, so if you can, please type in the chat box your answer. So, for the first question, which of the following are the start codon? If you answered A, then you're absolutely correct. Now, question number 2. Which of the following are the stop codon? If you answered all of the above, then you're correct. Now, last question. Question number 3. Which of the following is the amino acid that is linked with the start codon? If you answered threonine, then you are absolutely wrong. But it's actually methionine, guys. But we are finally on our last subtopic. As discussed earlier, that inside every cell is a hidden message, a genetic instruction manual written in letters A, T, C, and G. Today, we will learn how to crack that code. Let's start with a question. What is a genetic code? Uh, if you have no mic, please answer in the chat box. Okay, I can see so many answers now. So, thank you for your answers. But if you answered it is a set of rules defining how the four-letter code of DNA is translated into 20-letter code of amino acids, then you're correct. So, these letters, A, U, C, and G, are RNA nucleotides. They form words made of three letters each, which are called your codons, as you uh, heard earlier before. AUG, what is AUG again? Yes, of course, your start codon. And each codon instructs to build of a protein. So, to decode a message, we start with DNA, then transcribe it into mRNA, and finally match it with the tRNA during translation. So, let's learn how to use the codon chart to decode mRNA into amino acids. So, let's take a look at your mRNA code. Find the first letter on the base of the chart, okay? Then find the second letter across the top. Then afterwards, match the row and column to find the right box. Then finally, look for the third letter inside that box to get the exact codon. And that codon gives you the name of the amino acid. So I will give you some time to familiarize, the codon chart. Then afterwards, we will have a quick little activity. Okay, so let's try to decode this DNA strand. Okay, write it in your paper if you have. Then let's decode it into an mRNA and tRNA. When decoding DNA to mRNA, adenine rather is gained with uracil, thymine with adenine, and cysteine with guanine or guanine. So, what is your final answer? You can type it in your chat box. Okay, very good. That's correct, Mr. Calapre. Thank you so much, Mr. Calapre. Now, now that we're done with that, translation is more than just a cool process. It's how your body makes everything from insulin to hemoglobin. And in clinical settings, understanding translations help explain genetic diseases, like when a mutation causes a premature stop codon. This leads to an incomplete protein, which might not work at all, and that can cause serious health issues. Now, future medtechs, that is all for today, and thank you for listening.